Hello everyone, welcome back to my channel. So today I'm here to discuss about my TIFR Mumbai interview experience and what all questions I have been asked. So my interview timing was 2:40 p.m. on 24th of March, and as soon as I entered the room, they were like, are you nervous or not? So I was like, yeah, I am a little bit nervous, and they were trying to make me comfortable. And there were three interviewers first of all, but after I just introduced myself. One of them had their camera on and others just switched off their camera for my ease. So they started with the question that what is my area of interest, like what subject. So I answered it as molecular biology, as I am quite interested in molecular biology and cancer. And next question they had is what all subjects were there in your graduation. So I said I am a botany graduate and I read all the subjects like molecular biology, biochemistry, plant physiology and XYZ. I told all the subjects which I read, and I had chemistry and zoology as my elective, so I have told them. Now the first question which they had was very basic, that what do you understand by the term gene? So I answered that gene is a part of chromosome which is responsible for functionality of a trait or any character, which is correct according to genetics, but they wanted a definition related to molecular biology, which could have been a gene is a fundamental unit of hereditary which is made up of DNA in some and RNA in some viruses. It contains the instructions for building up the proteins. This could have been a better answer, but I don't know whether they were satisfied with my answer or not. Now after that, ma'am asked me a question that if you are given a 10 kilo base pair sequence, how can you say that a gene is present in that sequence? Now, as I was nervous, so I took a lot of time thinking about this. Now, doing experiments in lab, one of the best techniques could have been PCR to detect whether a gene is present or not. But if you are working with a computer, you want to go for bioinformatics, so there can be open reading frame technique, sequence annotation database, regulatory sequence prediction as well. Now after this question completed, other interviewer asked me a question that suppose you are given 4 test tubes, in which one test tube has bacterial cell, another has yeast, third one has insect and fourth one has human. How can you distinguish between all the 4, or how can you say that in which test tube which organism cell is there, given that you are provided with a simple microscope? So I started with giving a basic answer, that is I will first distinguish based on prokaryotes and eukaryotes. So it could have been easily distinguished between bacteria and other type of cells. So this was figured out on the basis of, and second I could have figured out on the basis of cell wall as well. Bacteria and yeast may have a cell wall while human and insects may not. Now next question was how can you distinguish between this human and insect cell when I have given you more equipments, like you can use staining techniques as well. So one of the staining which I said that I could use staining based on the hemocyanin found in insects and hemoglobin found in humans. So the staining property or the staining color which comes after that will tell me that what type of cell is it. But another thing which I could have told, that while staining the human cell with methylene blue, the nucleus will be very prominent because due to the presence of a huge nucleus. So it could have been a distinguishing features among them. So these were the questions asked during my TIFR interview. I was very nervous, but they were trying to help me in every step. They wanted to know my analytical skills rather than the fact-based skills. So I hope this would help everyone. Thank you so much for watching the video. Bye bye.